My name is Joao Pedras-Vasconcelos. I'm a product quality and immunogenicity reviewer for the Office of Biotechnology Products, Center for Drugs Evaluation and Research, Food and Drug Administration in the United States. I am a trained immunologist. My graduate degree was a PhD from Cornell University in cellular and immunology and immunoparasitology. So I already had an interest in immunology per se from my training. In addition, as part of my job, I've been with the Office of Biotechnology Products for 16 years now. And we, in the Office of Biotechnology Products, we do both product quality as well as immunogenicity reviews. And that comes as a result of historical circumstances and due to the nature of the products we regulate within the office. Many of our products have an immunological component, monoclonal antibodies, as well as cytokines, growth factors, and so they impact the immune system. And in addition, we also deal with a lot of what we consider high-risk products, such as enzyme replacement therapies for genetic diseases of the kind, such as lysosomal storage diseases. And thus, immunogenicity has always been at the forefront of our concerns, both because a substantial number of us are trained immunologists, and as well as due to the nature of the products that we regulate at the quality level. And because we understand part of the immunogenicity, understanding immunogenicity of a product has to deal with how do the critical quality attributes of a product, these are those factors inherent to the molecule, in my case biologics and sometimes drugs, how they impact safety and efficacy in the patients. And a lot of this impact of safety and efficacy is modulated by the patient's immune response to the product. So it goes very closely hand in hand, both the interest in quality as well as the interest in immunogenicity for the kind of products we regulate in the Office of Biotechnology Products. I wear both hats, right? And my hat as an immunogenicity reviewer, I'm considered a subject matter expert. I'm a current co-chair of immunogenicity working group for the Office of Biotechnology Products. So that places upon myself as a subject matter expert a series of requirements when we first get a submission. As an immunogenicity reviewer, first concern is when we get a submission is what's the risk assessment for this product in terms of what's the immunogenic potential for this product, and this kind of assessment involves examining roughly three broad questions. One is what are the molecular characteristics of the product that could impact immunogenicity, and then what are the patient factors involved. Basically, was the patient population involved? This product that's going to be exposed to this product, and how likely are they to produce an immune response to the product? And lastly, what are the factors in the clinical trial in terms of sampling plans and dosing that could lead to an immune response to the product? So these are broadly three categories that we look at. Furthermore, we also look at the validation reports for the immunogenicity assays. In fact, the Office of Biotechnology Products is responsible for examining the validation reports for not only biologics, large molecule drugs, but also small molecule drugs within the Center for Drugs Evaluation and Research. In addition, we collaborate with the clinical and ClinPharm reviewers, as well as the ClinStats reviewers, or clinical statisticians, in looking at the impact of that anti-drug antibodies may have developed regarding safety and efficacy data, meaning PK/PD data, as well as adverse event data. So this is a very collaborative process, but as you may be able to ascertain, it involves an analysis at multiple levels and throughout the life cycle of the product. So it's interesting because as a product quality reviewer, again, I have both roles, when you have a submission, there's something called the Electronic Common Technical Document that comes as part of the International Conference on Harmonization. The ECTD is the format, the electronic format, which documents get submitted. Now, if you're doing quality review, you have a module dedicated to you, and you have basically a linear sequence of analysis and linear sequence of review. But when you're doing immunogenicity, the information is very scattered in nature. It involves modules 2, it involves modules, various sections, module 5, and often it involves an upteenth number of reports of validation and clinical assessments that is very challenging to actually go through in a coherent fashion in a limited time frame that we have for evaluation. So that's really for a immunogenicity review within the Office of Biotechnology Products the biggest challenge. We're trying to, there is put forth, there is something called Integrated Summaries of Immunogenicity, which initially was put forth by your European colleagues that we really like, and the way that the recommendations were put forth in the 2015 guidelines. And we've been encouraging sponsors to follow that approach because we found it really cuts down on our review time and the difficulties of our review. So I speak this as these are my own views and not necessarily those of the agency. And what I hope and I believe is going to happen is there's sort of two broad areas. One is you clearly are developing assays or developing assays that allow for multiple levels of information to be obtained, not just simply the levels of anti-drug antibodies that you get or the levels of neutralizing antibodies that you get, but it may potentially information such as the isotype of antibodies that you get. And that may actually be quite important in determining risk for patients down the line. The second line of development I believe is going to happen is actually looking at earlier within an immune response for biomarkers that are not from the adaptive immune response, and not necessarily attached to antibody responses, but will subsequently play a role in priming those antibody responses. I am referring to signals from the innate immune system, early signals that we may be able to develop assays to pick these signals up that would allow much greater patient-to-patient understanding and potentially an early segregation of patients that may be more at risk of developing anti-drug antibodies versus those that are less at risk of developing anti-drug antibodies. So those are two broad areas that I believe we will see the future go within the area of immunogenicity. I love that question. I love that question because I wish I'd given that advice to myself 20 years ago. I think there's two things I've learned. I spent 20 years in the lab and I've been a reviewer for 16 years. And what I've learned is, my first advice is know yourself. Know what makes you tick. Know what drives you. Know what passions you have. Really, before even you continue careers, assess your own personality. There's a number of different tests out there to give you an idea of your personality. I think that's crucial. That's half the battle is knowing yourself and what works for you, what doesn't work for you. The second piece of advice, it's along the same lines, is take time for yourself. Take time to balance your life. Take time to take care of your inner health as well as your outer health. Because if you don't have that, doesn't matter how much you put into your work, it's not going to turn out well in the end. So those are the two biggest pieces of advice. If you're able to create an equanimity, a sense of equanimity, balance, realize that you're not your next experiment. Realize you're much more than your previous result. The fact that you had a bad result in the lab does not mean that you're a bad person or that you don't know what you're doing. So it's really being able to be kind to yourself through the challenges and to know yourself. I think those are two essential things pieces of advice I would tell any new scientist.